Hello, everyone. Welcome to today's seminar series called Cutting Edge Advances Towards Personalized Mental Health, hosted by our Center for Precision Mental Health and Wellness. I'm Dr. Xue Zhang, and I'm a co-director of this seminar series along with Dr. Leonardo Tozzi. I will kick off with a couple of housekeeping items. This webinar is being recorded and will be posted on our website next week. So you can find our website in the chat box or scan the QR code on the left side of the screen. From there, you can sign up to receive announcements about our future seminar series and events. If you're interested to join our center as a research member, please also find the link in the chat box or scan the QR code on the right side of the screen to fill in a survey, and we will get back to you. And for the talk, please put your questions in the Q&A box, and we will attend to those at the end of the presentation. I encourage you to put your questions as early as possible, as we will be addressing them in order. Now I hand over to our director of our center, Dr. Lian Williams, to introduce our special keynote speaker today, Dr. Katherine Narr.
Hello, everyone. It's wonderful to have you join the Cutting Edge seminar series. It really is a great privilege to introduce Professor Katherine Narr. I'm a big admirer of her work, as are many in our center, and thrilled to be a collaborator. Dr. Narr is a professor in the Departments of Neurology, Psychiatry and Behavioral Sciences at the Geffen School of Medicine at the University of California, Los Angeles, UCLA. She's also a faculty member of the UCLA Amundsen Lovelace Brain Mapping Center. And as you'll hear, Dr. Narr is right at the cutting edge of using imaging for precision therapeutics. She's recognized for her work using multiple imaging methods for the study of depression and other psychiatric disorders. And she's served as a PI of multiple major NIH grants. A recent U grant is a human connectome project investigating the perturbation of depression and its connectome using fast-acting therapies. A focus of Dr. Narr's ongoing research centers on using multiple neuroimaging methods as predictors and correlates of rapid-acting therapeutic responses in patients with depression. As you'll hear, she follows patients prospectively using electroconvulsive therapy, serial ketamine therapy, total sleep deprivation, and transcranial direct current stimulation. I've been a long-standing admirer of Professor Narr's work and glad to have the recent opportunity to collaborate on a joint project that will combine our human connectome data sets across centers and develop more robust and scalable predictors of response to these rapid-acting treatments, as well as subtypes of depression and its comorbid disorders. Today, Professor Narr will talk on imaging biomarkers and predictors of rapid treatment response following ketamine therapy in major depression. I'm delighted to hand over to her. Thank you.
Hi, everyone. It's my pleasure to be here remotely. And yeah, I want to thank Dr. Williams, Tozzi, and Zhang for inviting me, and look forward to hearing your questions. Just a quick question. So what time should I shoot for ending the talk? Did you say like 40 minutes, like 11?
Yeah, 40 to 45 will be perfect.
Okay, so like I should aim to finish at like 11:40.
That sounds good.
Okay, good. All right, good. All right, well, we can, I guess, get started. Let me share my screen again. Right. Can everyone see that?
Yes.
You good? All right, good. Okay, so Dr. Williams already mentioned that I'll be talking about imaging biomarkers of treatment response with regard to ketamine treatment in major depression. I'll spend less emphasis on talking about predictors today, but anyway, so here we go. So, whoops, I think I've already missed one. It's weird. All right, just as an outline of what I'm going to talk about today, just provide you some background of ketamine as an antidepressant treatment and the importance of using multimodal MRI for understanding ketamine's therapeutic effects. I'm going to focus on describing some resting state study findings of ketamine response in major depression and a summary of these findings, some work using arterial spin labeling, also talk about task fMRI findings and provide a summary of these findings in relation to ketamine treatment in major depression. We'll touch on some limitations of the ketamine imaging field to date and some avenues for potential future research might be important. All right, so this group is already well familiar that major depression is the world's leading cause of disability, that's days lost to work, that depression is clinically heterogeneous and involves multiple different symptoms that span multiple domains. And it's a serious burden to the healthcare system in the US and across the world. Okay, luckily, depression is treatable, but unfortunately, as we learned from the landmark STAR-D trials, standard antidepressant monoaminergic pharmacotherapies are only moderately effective. Half of patients fail to remit to their first initial treatment with standard antidepressants, and 30% of patients fail to respond adequately to 2 or more antidepressant therapies of adequate dose and duration. These patients are referred to as having treatment-resistant depression, and there's even a higher burden of suffering for people with TRD, as well as healthcare costs. For example, there's a higher risk of relapse. We know that there's an 80% recurrence of having another episode after a person has had more than 2 or more major depressive episodes. The depression becomes more intractable with time. Healthcare costs are about 20-fold greater. There's higher morbidity and mortality. TRD has higher suicide risk. And the patients also have more variable response to alternative treatments. Another downside of standard antidepressant treatments, as shown in this schematic, is that response is relatively protracted and takes weeks to months for patients to achieve adequate clinical benefits. This highlights the need for more effective, personalized and rapidly acting treatments. Currently, ECT is considered the most effective FDA-approved rapidly acting treatment for major depression, but ECT also has some downsides. There's stigma, anesthesia is required, there's potential for cognitive side effects. So this is where ketamine comes in, and here, this figure from Zhang, Yao, and Hashimoto gives a timeline of the discovery of ketamine, its first chemical synthesis in 1962, its approval for use as an anesthetic by the FDA in the 1970s. I just wanted to note, sorry, I keep doing that, that ketamine is a racemic mixture of RNS enantiomers. So ketamine, when I talk about ketamine, I'm referring to racemic ketamine unless I'm specifically talking about S-ketamine or R-ketamine. For depression, the first placebo randomized controlled trial for ketamine at low dose for use as an antidepressant was by Berman and colleagues in 2000. These results were replicated in 2006 by Carlos Zarate and the NIMH group. Since then, there have been numerous additional randomized controlled trials and studies. In 2019, S-ketamine, so the S enantiomer of ketamine, received FDA approval as an antidepressant, which has a nasal administration in the US and USA in Europe. I just want to note that racemic ketamine does not have FDA approval for specific use as an antidepressant, although is widely used off-label for this purpose. R-ketamine, there's also a timeline for R-ketamine. Preclinical models have shown promise that R-ketamine might be an effective antidepressant, and recently R-ketamine received FDA IND clearance for clinical use. So watch that space. All right, so ketamine, as I mentioned, many clinical trials and open label studies have shown that ketamine has these robust antidepressant effects. Generally speaking, about 20% of patients, and these are patients typically with treatment-resistant depression, remit after just a single dose of sub-anesthetic ketamine, and 50% respond. Ketamine has a very rapid effect on mood, beginning within hours. The peak reduction in mood is generally at about 24 hours post-ketamine infusion. Ketamine also has a high record of safety, as we know from its decades of use at higher doses as an anesthetic in medicine. The graph shows mood scores from a group of TRD patients from our own work who received ketamine treatment, 4 ketamine treatments, and the graph shows Hamilton Depression Rating Scale means at baseline before ketamine, after 24 hours after a single dose of ketamine, 24 hours after 4 ketamine treatments, and then 5 weeks after receiving ketamine. And as you can see, there's very pronounced reductions in mood, even after 1 dose, which are increase in magnitude after serial ketamine, but then start returning towards baseline after a number of weeks. Ketamine also has these rapid effects on reducing suicidality, and has also been shown to have pronounced anti-anhedonic effects that appear independent of mood. This is important because standard antidepressants are, anhedonia is less responsive to standard antidepressants, let me put it that way. The graphs below show the same study design prior to ketamine, after 1 dose of ketamine, after 4 doses, and at follow-up for suicidality, symptoms of apathy, and anhedonia. Here I will mention, as I didn't before, that typically racemic ketamine is dosed at 0.5 milligrams per kilogram for a period of 40 minutes. There have been some dose-finding studies, and this does appear to be the most effective dose for ketamine's antidepressant effects. Okay. So it's less widely known that sub-anesthetic ketamine actually seems to have these pro-cognitive effects and positively impacts certain aspects of cognitive function. This is again data from our group. Here we used measures from the NIH Toolbox cognition battery, which you can see in the graphs below. And we found that over the course of treatment, the flanker inhibition, which is response inhibition, list sorting, processing speed, picture, sequence, and vocabulary all increase significantly from baseline to the end of 3 treatments, but also tend to keep increasing into the 5-week follow-up. We did mediation analysis, and it seems like these pro-cognitive effects are mostly independent of mood, which sort of implies that, ketamine targets not only depression-relevant brain circuitry, but also brain circuits related to these different cognitive functions. Okay, so unfortunately, ketamine is not the answer to treating depression. It has several important limitations. Most critical is that ketamine antidepressant effects are relatively transient. For one dose, response typically starts to diminish within a week, heading towards relapse. However, serial treatment does improve and extend response, but again, relapse usually occurs within weeks of treatment cessation. Maintenance treatments have been shown in recent clinical trials to help sustain response, and there was a recent clinical trial of ketamine published that included computerized self-association, sort of affirmation training that showed that it prolonged ketamine response beyond the actual infusions. Not everyone is eligible to receive ketamine for medical reasons, particularly people with cardiovascular disease and hypertension. Ketamine is classified as a Schedule III drug of abuse and is certainly used recreationally and has a lot of different street names. Certainly at high doses, ketamine is associated with cognitive problems, but people who are at risk for abuse for psychoactive substances might not be good candidates to receive antidepressant ketamine therapy. Ketamine has psychotic effects at high doses and dissociative and psychomimetic effects, even at sub-anesthetic dose. However, usually these effects diminish within hours of the treatment itself. S-ketamine has higher NMDAR binding affinities and is more subjective to these psychomimetic effects. Finally, little is known about the long-term safety of using sub-anesthetic ketamine for the treatment of depression. So more work needs to be done just to make sure that it doesn't have long-term side effects. All right, so challenges that remain for the translation of ketamine to full-scale clinical use, although it's certainly used already in some contexts through S-ketamine, which is really expensive, and racemic ketamine, which is off-label. But one of the primary challenges is that the mechanisms of ketamine's antidepressant action aren't fully understood, and neither are the contributions of individual factors to how particular patients might respond to ketamine. As we mentioned, the heterogeneity in depression, demographic and clinical factors, although one of the predictors of treatment response to ketamine in pretty much all antidepressants is treatment resistant. People who are treatment resistant are less likely than other patients to respond to further treatments. understanding medical psychiatric comorbidities and their contribution, cognitive and behavioral characteristics, how specific genetics might impact ketamine response, environmental factors. So there remains a need for viable biomarkers to understand response mechanisms in ketamine. Since this talk is focused on imaging, I'm not going to thankfully dwell long in this slide. It is from a review article from Zanis and Gold that illustrates the large amount of work that's been done in the space of trying to determine the cellular and molecular mechanisms contributing to ketamine's antidepressant response. And this is not completely resolved, and nor are these different mechanisms that have been proposed, inhibition of glutamate in GABA interneurons, for example, versus inhibition of NMDR lateral habenular neurons. These are not necessarily independent. I mean, they might be complementary, but the bottom line is that we still don't know exactly what the mechanisms are of ketamine's antidepressant action at the molecular and cellular level, although we understand that these effects might be NMDR or NMDR independent, so downstream of NMDR antagonism, and also that these blockade of the NMDA receptors by ketamine lead to this cascade of other processes, including increase in BND, brain-derived neurotrophic factor, and initiation of the MTR system of protein synthesis, and also affect monoaminergic neurotransmission, as well as the opiate system appears to be involved as well. Okay, so I already said more than I planned to, but yeah, complicated. Okay, irrespective of what happens at the molecular and cellular level, we still need to understand how ketamine modulates functional brain systems to understand how perturbation of these brain systems impact behavior. So, you know, whatever happens, these are upstream of what happens at the level of the synapse, but ultimately ketamine acts in some way to change brain circuitry, and that in some ways is expected to relate to antidepressant response. So it's important. Certainly human imaging provides a critical opportunity to address that question that might also help us understand what's happening at the molecular and cellular level. Looking at imaging biomarkers is also important because it might help us identify predictors of risk, response and relapse, which can inform more personalized treatment and prevention strategies and guide novel treatment development. Okay, I'm not going to dwell on this slide either. This is an oversimplistic model of depression circuitry. Sorry, keep on doing that by accident, showing sort of brain systems that are involved in major depression, that might involve an underreactive top-down set of brain regions and networks, and an overreactive bottom-up. It shows how different imaging modalities can help us infer various mechanisms that might help us understand treatment response. Okay. So now I'm actually going to start talking about imaging data. So from this point on, I'm going to summarize some recent research concerning how ketamine modulates brain function and connectivity and metabolism in major depression. When I was planning this talk, I was planning to talk about all the different imaging modalities. It's just way too much. So I'm going to focus this talk on discussing findings using resting state fMRI, task fMRI, and also present some work using perfusion imaging. I'm not going to talk about PET imaging. Most of the PET studies to date have been focused on looking at the acute effects of ketamine. So ketamine, what happens to metabolism, when the patient is actually during ketamine treatment or shortly after. All the work that I'm going to discuss today is focused more on the neuroplasticity that happens after ketamine. So after the drug has been metabolized from the system, so 24 hours and later. There has been some work with diffusion MRI, including from our own group, and structural MRI. I'm not going to talk, there are much fewer studies. I'm not going to talk about that work. Magnetic resonance spectroscopy, there have been a number of studies. These have been a little disappointing because they haven't, at least at beyond the acute effects of ketamine, there haven't been any promising, biomarkers. One would expect increases in glutamate, but that is not the case after after ketamine has already been metabolized. Okay, before I talk about the data, I'm just going to take a sidebar to describe our connectome related to human disease UO1 project, because when talking about resting state and task fMRI, I'm going to punctuate existing findings with our own data, so just so that you can get an idea what how our ketamine study was designed. So this connectome related to human disease project is obviously has different goals, but is the same as Dr. Williams's, same sort of general structure as Dr. Williams's connectomes related to human disease project. Okay, so the goal of this NIH-funded UO1 that was completed about a year ago, and the data is about to be shared any day now through the NDA for anyone who's interested, was to understand how faster-acting treatment modalities that have different access to the central nervous system through neurostimulation, pharmacotherapy, as well as behavioral, so that included ECT, ketamine, and total sleep deprivation, affect the functional and structural connectome in major depression. We used the human connectome setup. We scanned people before and after treatment. We also included some intermediate time points for ketamine. Patients were extensively clinically and cognitively phenotyped. We also included, we collected blood for gene expression and to look at inflammatory biomarkers. Okay, but today we're focusing on ketamine. This flow chart illustrates sort of what happened in our study. All patients enrolled were defined to have treatment-resistant depression. They were between 18 and 65. We included bipolar and unipolar as long as they were in the midst of a major depressive episode that had to last for at least 6 months. We included patients who were taking standard antidepressant medications. We didn't, if patients were taking any benzodiazepine, they were asked to not take those during the study. We excluded substance abuse, people who were imminently suicidal, although most patients had suicidal ideation, and obviously dementia, schizophrenia, any neurological disorders. Patients were scanned within a week of receiving serial ketamine infusions which were administered 3 times a week, for a total of 4 infusions. they received an MRI 24 hours after the first infusion. This was actually not a UO1 study point and was supported by intramural funds, and we have a slightly smaller sample size there. But then after they received another MRI 24 hours after their fourth infusion, and then after 5 weeks in follow-up. Okay. Here is just a summary of our multimodal MRI, structural MRI with high spatial resolution, 0.8 millimeter isotropic resolution, diffusion, 185 gradient directions, perfusion imaging, which use multiband, 2.5 millimeter isotropic. Task fMRI we chose. It was a clinical study and there was a lot of burden of patients. We restricted the task MRI to 2 tasks, emotion regulation and response inhibition, and we'll talk about this later. And then finally we did resting state fMRI using HCP protocols, multiband, 2 millimeter isotropic, spatial resolution. Okay. All right, so before I talk about resting state results, with regard to ketamine treatment, I just wanted to remind you that there have been numerous studies that show that major depression is associated with dysfunctions in functional connectivity between and within large networks. Some networks are particularly well implicated, such as the default mode network, as well as the salience network, frontal parietal / central executive network, the ventral medial and orbital affective networks, limbic networks. Changes in functional connectivity have been shown to associate with treatment outcomes for other treatment modalities. The figure on the left that you're looking at is work from PNAS from Charles Gan et al., published in 2019. This study included, it was a cross-sectional study, included 1,300 patients with major depression and over 1,000 healthy controls. And the purpose was to look specifically at default mode connectivity, differences between patients and controls. And they found in this large sample size that there was decreases in functional connectivity within the default mode network, which was impacted by the recurrence of depression as well as medication. They also looked at other major networks as defined by the 7-EO atlas and found decreases in connectivity within the visual and somatomotor network, and as well as between them, and the dorsal tension network. Okay, all right, that's just a background to emphasize depression is associated with changes in functional connectivity. All right, perhaps not surprisingly, most of the studies published to date in the last 10 years to evaluate functional connectivity changes in association with ketamine have been resting state studies. There are about 15 studies to date, and about 3 of these studies have looked at serial ketamine. A number of these studies actually include the same sample, so it can be a little bit confusing. In general, as you are probably all aware, there are many different ways of looking at functional connectivity, despite using different methodologies. Certainly the take-home is that ketamine does perturb functional connectivity in and across large-scale networks. And perhaps the default mode network and its connections have been mostly implicated, but also probably mostly studied. Here on the left and the right, I'm just illustrating, for example, results from 2 different studies, one from Siegel et al., where they looked at connectivity between the subgenual anterior cingulate and large-scale networks, including the default mode network. Well, actually, they focused on the default mode network. And they found that after ketamine infusion, and this ketamine infusion was somewhat different because they did a ketamine infusion over 96 hours in their patients, and the scans were collected 2 weeks follow-up. But nevertheless, they found a decrease in connectivity between the subgenuine cingulate and the default mode network following ketamine in patients with depression. Another study by Evans et al., this is the NIMH group, found increased default mode anterior insular connectivity 2 days after treatment. And this is what is illustrated in the figure. Patients were also scanned. It was a crossover study, but at 10 days after they received treatment, these effects were no longer present. All right. This is a study by our own group, given that the limbic system is so, well, it's more than implicated in depression. That a lot of imaging studies support disturbances in limbic function and structure in major depression. We wanted to look at connectivity from these limbic regions, specifically the hippocampus and amygdala, to large-scale networks focusing on the rich 3, I guess, the default mode, central executive, and salience network. And to do this, we used a seed-based approach. We used the Harvard-Oxford Atlas, I believe, to obtain hippocampus and amygdala ROIs, and then we use group ICA and dual regression to extract these 3 networks. The central executive was split into left and right. And we found that treatment with ketamine, single and after 4 treatments, associated with increased amygdala right central executive connectivity, and ketamine associated with decreased amygdala salience network activity. We also found that ketamine increased hippocampus left central executive resting state functional connectivity. And these results are illustrated here. HC stands for controls, T1 to T3, it's baseline after first treatment, after 4th treatment. But what's interesting is that these changes in connectivity between baseline and the first treatment associated with improvements in anhedonia, and changes in between amygdala and salience network connectivity associated with improved anxious anxiety as measured by the SPAS scale. Sorry, just checking the time. Gosh, time goes so quickly when you're having fun. Okay. All right. Well, at the same time, another way of looking at functional connectivity that is not biased by any a priori hypotheses using seed-based or single network approaches is to use a data-driven connectomics approach. And we did apply this approach to our data. And here, what we did was we used ICA, high-dimensional ICA, to functionally segment our resting state data and into nodes, and to look at connectivity or the edges between all of those nodes. A benefit of this approach is that, yeah, it's completely data-driven, and so it is not biased anyway. A disadvantage is that functional segmentation requires large data sets, either a large amount of, large duration of resting state or a large sample. Results from this study showed that connectivity between the cerebellum and the dorsal salience network decreased following serial ketamine treatment. We also found an interaction with remitter status. So I didn't actually note this, but I did tell you that most patients respond to ketamine. And in our sample, by the end of treatment, about 50% had completely remitted to their depressive symptoms. So when binarizing, we typically binarize by whether a patient remits or not, rather than response, which is generally defined as a reduction by 50% or more in mood scales. In any event, this increased cortical striatal cerebellum connectivity associated with change in Hamilton Depression Rating Scales over time. We also found that ventral attention to visual network and default mode to visual network functional connectivity that was higher in patients versus controls tended to normalize with treatment. So this sort of emphasizes the importance of visual networks, which are usually not focused on, but are usually there in the results, even in cross-sectional studies. Okay. All right. So, well, data-driven connectomics is really wonderful and important. There are reasons why you might want to really focus in on a specific region. For example, the lateral habenula. The lateral habenula is an important structure in the brain for its role in reward, fear, and decision-making. Activation of lateral habenula neurons occurs when, in the absence of reward, and inhibition, the lateral habenula is sort of referred to as the anti-reward center. Didn't explain that very well. There's been critical preclinical work that was published in Nature in 2018 that showed that increased burst activity in depression animal models is reversed by ketamine, and blockade of lateral habenula neurons by an MDAR antagonism is sufficient to induce antidepressant effects. One group has looked at habenula to cortical connectivity following single dose ketamine, and they found that reduction in mood scales measured by the Montgomery-Åsberg rating scale was associated with increased right habenula, right frontal pole functional connectivity, and reduction in subjective ratings of mood was associated with increased right habenula, occipital pole, temporal pole, parahippocampal gyrus, and left lateral occipital cortex. Yeah, okay. We were also interested in looking at habenula connectivity in our study, and this shows our processing pipeline. We segmented the habenula using the methods of Eli et al. 2019. We looked at connectivity from the habenula to all voxels in the brain. You can see the resting state connectivity map shown at the bottom. We didn't exactly replicate. Our results were somewhat consistent. Well, yeah, maybe that's. We found changes in connectivity between the left habenula and with visual cortex. We found there was a difference in connectivity between remitters and non-remitters of ketamine, as shown here. Changes occurred in the same direction, but much larger in remitters. We also found that connectivity of the left habenula with the posterior cerebellum, so the cerebellum keeps on coming up, at baseline predicted clinical outcome measured with the HDRS. Okay. All right. It looks like I don't have that much time. Okay, very briefly, with our ASL data collected using human connectome protocols, we looked at differences in perfusion that occur with single and serial ketamine, and we found after single ketamine that there were reductions in increases in perfusion in visual association and primary visual regions that are associated with antidepressant response. Interestingly, after serial ketamine, we found decreases in hippocampal perfusion, which were bilateral. As well as the anterior insula. All right, summary of resting state. Yeah, there have been a lot of different study designs and analysis approaches and small samples that are overlapping. So it's not surprising that coming up with a coherent story of how ketamine perturbs functional connectivity is still rather challenging, but results do in general suggest increased functional connectivity between large-scale networks, in particular the default mode network, frontoparietal network with nodes including the anterior insular striatum, amygdala, and habenula. We found decreases in functional connectivity in the cerebellostriatal cortical network, as well as other network components. The connectograms you're looking at are taken from a recent review that my postdoc published in Biological Psychiatry, CNNI. It includes very nice tables of all the resting state fMRI studies using ketamine published to date. And anyway, this connectogram is color-coded mapping to networks shown on the surfaces of the brain to sort of summarize what the existing resting state studies of ketamine have shown. Okay, task-related fMRI studies. There have been fewer task-related fMRI studies using ketamine, about 9 studies. These studies have investigated tasks of reward, emotion recognition, and judgment, and response inhibition. Again, have included, even in the 9 studies, have included overlapping samples. Here are results from Morris et al. That they used a reward incentive processing task focused on looking at subgenual anterior cingulate activation. They found this was increased in major depression for trials of positive or negative feedback, and they found that that ketamine reduced this hyperactivation. Gosh, I think it was for positive or negative feedback trials, and baseline activation correlated to improved anhedonia. Okay, as I mentioned earlier, we performed 2 different tasks in our UR1 sample. We chose the motion recognition task because it produces quite robust amygdala activation. Here you're looking at an example of the trials of the task and the average activation map showing the whole face processing network, as well as bilateral amygdala activation. When comparing groups, in this study, we actually compared controls and patients who received ECT and ketamine, and we found that both patients receiving ketamine and ECT, here we just focused on looking at the end of serial ketamine because we looked at the end of the ECT index. So time point 2 here is after 4 ketamine treatments. In any event, we found both patients receiving ECT and ketamine have reduced responsivity of amygdala activation for fearful and happy faces relative to objects. We also found in the larger face processing network that there were significant relationships between activation for fearful or happy faces with change in mood, reduction in anxiety, and change in anhedonia. The regression lines are shown separately for ketamine and ECT patients and were more robust for ketamine than ECT, particularly for anhedonia. The second task we looked at was response inhibition using the carrot go, no-go task. This task was used because it's a proxy for the cognitive control network that's been shown as disturbed in depression. Here at the bottom, you can see average activation maps for controls, patients, and controls and patients together. For this task only has 2 contrasts, the go greater than no-go and the no-go greater than go. And we interested in the no-go greater than go, which is the response inhibition condition. Interestingly, with ketamine, there were pretty robust changes in this cognitive control network, as illustrated by this activation map, but also by the graphs below for different regions in this activation map, showing controls in blue, and then after baseline, first and fourth treatments. Changes, also these changes in bold signal also associated with reductions in mood scores as measured by the HDRS, and some clusters associated with reductions in rumination following serial ketamine. Okay, we followed up this study because we were specifically interested in looking at functional connectivity between the cerebellum during response inhibition and these the frontal parietal salience and somatomotor networks. So we did a follow-up PPI analysis, and we found that there are these relationships in connectivity between certain regions of the cerebellum to the frontal parietal network that differ by remitter status and also relate to the extent of change in subjective mood ratings. Here is a figure illustrating results, summarizing results from the functional task fMRI studies that have looked at connectivity. I see that's a typo. It's not changes in resting state. It's task fMRI. This is also from the recently published review I mentioned, and shows the direction of changes in activation, for example, for the response inhibition, the frontal parietal network, decreases in hippocampal, amygdala activation for emotional face recognition. So those are summarized here. This is a schematic illustrating, sort of attempting to summarize some of the major findings from resting state and task studies to date. It's a model, and we'll see how this changes over time, but I'm not going to spend too much time going into it. All right, so what are the limitations of all the work that's gone on to date? Well, of course, you know, it takes time to develop theories about and facts about how certain treatments impact functional systems. And, you know, up until now, sample sizes have been relatively small. Well, they have been small. Study designs have been different. Some have been randomized controlled trials, some have been open label. The timing of assessments has been different. Sometimes the dose of ketamine has been different. Depression is heterogeneous. People have included bipolar and unipolar depression, but also looking at other clinical populations. Another limitation is the quality and variability of image acquisition and analysis methods. For example, it's really important to obtain, you know, sufficient duration of resting state fMRI with, you know, limited motion artifacts. And certainly there's so many different ways of looking at functional connectivity that even when results seem to conflict, they might not actually conflict. So this underscores that this is really important need for systematic replication, and also for the use of, I think, more data-driven cross-validation in larger samples to impact, to make an impact on how ketamine is used in the clinical setting, and to inform the development of new treatments. But I don't want to de-emphasize how important the work is that has been published so far, that emphasizes that changes in functional connectivity, perfusion, and brain activation relate to improvements in mood, anhedonia, and other functional dimensions of depression. So where should future studies go beyond, you know, larger sample sizes and replication, and I think multi-site studies, is to look at comparing biomarkers across different fast-acting therapies. There's a lot of interest in psilocybin and other psychedelic treatments. So our group is interested in comparing S and R ketamine to look at functional systems. We, as I mentioned, collected gene expression data, and we finally have had that RNA sequencing data processed. So we're hoping to combine that from peripheral blood, which is a disadvantage, but nevertheless, to combine that with our imaging biomarkers to try clarify and expand understanding of ketamine's antidepressant effects. Finally, given this ketamine has these pro-cognitive effects, I think it's important that further studies look at the functional systems involved in changes in cognitive function. Also, it's important to not forget about evaluating the safety of ketamine in long-term contexts. All right, I have so many people to thank, to acknowledge for the work I showed you today. I've had some really, we have really fantastic, have had and currently have postdocs that have come through the lab who've led much of the work that I've shown you today. Of course, funding sources are always important. NIMH, thank you, and as well as intramural funding sources. All right, thanks. I went a little bit over, but I think I'm okay.
Thank you so much, Dr. Narr. This is really such a complicated study, and thanks for sharing all the up-to-date findings. And definitely, it seems ketamine involves effects among multiple large-scale networks, and definitely warrants future studies to systematically replicate. I realize we are running out of time. I can see we have one question in the Q&A box. I think you already addressed it to some degree in your talk, but do you want to probably quickly emphasize the association between the changes in brain activity and ketamine response and cessation of response?
Okay, so I think that's also something that hasn't really been addressed in existing studies is, yeah. Well, certainly after ketamine, it's important to also predict relapse. And this is true not of ketamine, but also other fast-acting treatments like ECT, where relapse is actually within a year about 50% of the time. Of course, it happens much quicker with ketamine. So I think looking at biomarkers that predict relapse is really critical. But this really is an important area that hasn't really been fully investigated, I think. We do have our 5-week follow-up data, but we haven't really focused on looking at that. And also we did have some attrition, so at the 5-week follow-up, but that's a really important question. Yeah.
Thank you. Again, we realize we're running late, and people, if you have any further questions, feel free to send it to us. And I want to ask Dr. Narr, are you open if we summarize all the questions we received and send it to you, if you could address them afterwards?
Of course, and I'm sorry I went over. I guess I thought I had more time. I had my phone next to me, but yeah, hopefully you found that interesting. I know it's a complex story, and I don't know, maybe with more time it would have been. I think it's going to, I think over time we'll get a clearer picture of what's going on. But, you know, I know Dr. Tarzi and Dr. Williams know this complicated resting state analysis story. It's sort of like, you know, you don't, yeah, always, you're not always measuring the same thing. So it's, yeah.
We
But
have thanks in the audience for just walking through this field, because you're obviously been such a leader in it, Katherine. And as you say, it's complicated. So I know that the audience who are staying on, they're still here with us. Really appreciate you taking us through. Yeah, it's a tour de force of doing this work with imaging and new treatments and answering all the questions about response, remission, cessation of response. It's difficult, and then to standardize the measures.
yeah.
Thanks for paving the way.
You're welcome. I think, yeah, we need biomarkers, but we need robust biomarkers. So we have to try and get resting state into a robust biomarker. Certainly the brain activation studies are just so much easier to interpret, and when there's strong signal changes, that's a biomarker. So yeah, anyway, sorry, I'm just going off on a tangent. All right.
We could have a long chat about this, but I'll hand back over to Shui, who'll wrap up, and just want to express my thanks for a wonderful presentation and taking us all through. And we're getting lots of thanks here in the Q&A. And yeah, back to you, Shui.
Yeah, so I was going to mention that we get a lot of appreciation in the Q&A, and I'm pretty sure people will ask questions afterwards, and we'll be sure to summarize them. So thanks to everyone for joining us in our today's seminar. I wish everybody stay safe and dry. So we'll see you next time. And thank you, Dr. Narr, for presenting this time.
Thank you very much. Thank you.
Thank you so much.